Magna, Graecia. Okay. And from Catanzaro, Italy. And the presentation is the stem cell associated transcription cofactor, ZHANF521, contributes to AF9 dependent hematopoietic stem cell transformation in acute myeloid leukemias by altering the gene expression landscape. Thank you. Please take your time. It's right, and give you left some minutes for the discussion.
Nice to give me the opportunity to present my data about the role of zinc finger-1 protein in acute myeloid leukemias sustained by the translocation 911. Hematopoiesis gives rise to blood cells of different lineages through normal life. Alterations in a normal developmental program for blood cell formation results in a variety of hematological diseases, including leukemia. Acute myeloid leukemia is an heterogeneous disorder characterized by the clonal expansion of myeloid progenitors in bone marrow as well as in peripheral blood. AMLs progress quickly and aggressively because the maturation block results in the accumulation of immature malignant cells and in the fewer number of completely differentiated cells. Many acute myeloid leukemias are characterized by chromosomal rearrangements involving the mixed lineage leukemia gene, normally required during hematopoiesis. MLL is a protein, is a component of a protein complex leading to the epigenetic transcriptional activation of OXA genes family. OXA genes are required for the self-renewal of hematopoietic stem cells, as well as for the expansion of immature progenitors pool. MLL gain of function mutations caused by chromosome rearrangements aberrantly enhance cell proliferation and suppress senescence of hematopoietic cells, causing leukemia development. One of the most frequent MLL rearrangements is the translocation 911, resulting in the fusion protein MLL-F9, which is considered an initiating factor for leukemogenesis. Zinc finger-1 is a stem cell-associated transcription cofactor characterized by 30 crop-like zinc finger motifs and an amino terminal motif able to bind the nucleosome remodeling and histone deacetylase complex. Zinc finger-1 protein is the ortholog of the murine protein EV3 and the paralog of the human protein zinc finger-3, with which it shares a degree sequence of homology. Zinc finger-1 is known to play a central role in maintaining the immature compartment of hematopoietic, mesenchymal, and neural system. It was described as a negative regulator of adipocytic and osteoblastic differentiation from human adipose-derived mesenchymal stem cells, as well as can induce neuroblast differentiation from embryonic stem cells. In normal hematopoiesis, zinc finger-1 is highly expressed in CD34 positive cells, especially in the more immature subset of CD133 positive cells, and its levels decline when progenitor cells are induced to differentiate into myeloid or lymphoid cells. Similarly, in mouse system, high levels of EV3 protein was detectable only in hematopoietic stem and myeloid progenitor cells, compared to effector cells. So in order to investigate the potential correlation between zinc fingerprint1 expression and specific gene rearrangements or mutations, MEL gene expression profile datasets were interrogated, and high levels of zinc fingerprint1 mRNA correlated with the translocation 911, as well as the translocation 712, as well as by rearrangements of chromosome 16. On the other hand, low levels of zinc fingerprint1 were observed in MLs expressing AML1 ETO and PML alpha fusion protein deriving from the translocation 821 and 1517 respectively. In addition, high expression levels were correlated with MLs bearing NRAS or KRAS mutations, as well as isocitrate dehydrogenase 2 mutation and EV1 mutation. Conversely, low expression levels for zinc fingerprint1 expression were observed in MLs with NPM1, CBP alpha, and FLT3 mutations. In addition, among Fab classification, high levels of zinc fingerprint1 mRNA were observed in N0 subgroup, correlating with a minimally differentiated myeloblastic leukemia with a poor prognosis. So, in order to investigate the potential role for zinc fingerprint1 in maintaining leukemic stem cells with MLL rearrangements, we transduced CD34 positive cells with an antiviral vector carrying MLL9 fusion gene alone or in combination with zinc fingerprint1. Transduction efficiency was evaluated by FACS analysis for HFP expression, resulting of about 50% of positivity in transduced cell populations. Zinc fingerprint1 transcript levels were significantly increased in zinc fingerprint1 overexpressing cells, as well as in MLL9 transduced cells, indicating a role for this oncoprotein in inducing zinc fingerprint1 gene expression, as you can observe in panel C and in panel E. Similarly, MLL9 transcript levels were assessed by RT-qPCR in single and double transduced cells, as you can observe in panel D. During cell culture expansion, the number of zinc fingerprint1 overexpressing cells was significantly increased compared to the control. MLL9 fusion gene was able to immortalize CD34 positive cells, but something noteworthy is that the cells got transduced by both zinc fingerprint1 and MLL9 fusion gene led to a further increase in cell proliferation. In addition, the number of hematopoietic progenitors was potentially enhanced in the cells co-expressing both two factors, especially in the long period. Then, gene expression profiles of zinc fingerprint1 and MLL9 overexpressing cells were performed by Cladium DLI at 23 days, whereas the double transduced cells were analyzed both at 23 and 58 days. Hierarchical clustering analysis showed a more similar pattern for single and double transduced cells at early time point compared to the double transduced cells in the long period. Venn diagrams illustrate the number of up or down regulated genes in MLF9, in MLF9 and zinc finger-1 cells, and at the early time point, and the double transduced cells in the long period compared to the control cells. In particular, we analyzed 136 upregulated genes and the common 163 negatively regulated genes. But in order to investigate the molecular pathways specifically related to zinc finger-1 and MLF9 combination, gene set enrichment analysis was performed, and several genes associated to cell cycle, proliferation, hematopoiesis, and cytokines were found. In particular, heatmap in panel A showed the known MLF9 target genes. For example, MIF2C, MLLT3, MLLT11, IGF2BP2, and MICE1, including also zinc finger-1. These genes were induced in all transduced cell populations, but especially by MLF9 fusion gene, indicating the reciprocal transcriptional feedback loop between zinc finger-1 and MLF9 gene. Some of these genes were validated by RT-qPCR in panel F, as reported in panel F. Among the well-known MLF9 target genes, there were two groups of genes. The first one negatively regulated by the double transduced cells in the long period, and the second one upregulated by the same cells in the same period. Interestingly, zinc finger-1 overexpression correlated with high amounts of ZB1 and epithelial to mesenchymal transition transcription factor. Similarly, new additional MLF9 target genes were found negatively or positively regulated by the double transduced cells in the long period, as you can observe in panel D and in panel E. But to evaluate the effect of zinc finger-1 and MLF9 combination in our system, we focused our attention on genes involved in promoting and maintaining MLF9 leukemic phenotype, including genes with epigenetic functions, cell cycle genes, and transporter genes. Regarding the first group, here new additional genes with a variety of epigenetic functions were found upregulated by the double transduced cells in the long period, including SETB1, zinc finger protein 57, RPS6K5, SMID3, ATLX, and KDM7A. But considering the significant increase in cell proliferation upon zinc finger-1 and MLF9 combination, we observed the upregulation of 6 cell cycle genes, including CDCA7, GES5, IGF-BP2, CDC14B, and BBX, and we observed also the downregulation of 4 apoptosis genes, including BCL2A1, MYC-T1, CDK15, and DATK1. Interestingly, TV transporter genes were found upregulated only in zinc finger-1 overexpressing cells, compared to MLF9 alone, suggesting a potential role of zinc finger-1 in cancer metabolism. To consolidate our data, we sequenced zinc finger-1 expression in CD34 positive cells immortalized with MLLF9 fusion gene using two specific zinc finger-1 shRNAs. Gene silencing was confirmed by RT-qPCR and immunoblotting, as you can observe in panel A and in panel C. And zinc finger-1 protein expression was almost completely abrogated by both shRNAs compared to the control, whereas MLLF9 transcript levels were unaffected. Interestingly, zinc finger-1 gene silencing led to a significant decrease in cell viability of leukemic stem cells, as you can observe in panel D. Additionally, we used the THP1 cells cell line, carrying MLLF9 translocation and expressing high amounts of zinc finger-1 protein. Zinc finger-1 gene silencing in THP1 cells led to a significant decrease in their proliferation and growth rate, as well as in their clonogenicity, as you can observe in panel F and in panel G. We confirmed this data using a second hematopoietic cell line, the MOLM13 cells, co-expressing both zinc finger-1 protein and MLLF9 fusion protein. In this case, we used as a negative control the negative zinc finger-1 HL60 cells. Also, in MOLM13 cells, the clonogenicity capacity was significantly reduced by zinc finger-1 gene silencing. So our data indicate a strong cooperation between zinc finger-1 and MLLF9 in the development, in the maintenance, and in the clonal expansion of leukemic cells, leukemic stem cells. So, in conclusion, the majority of AML patients were positive for MLLF9, also express levels of zinc finger-1 protein, correlating with a poor prognosis. By performing gene expression profiling of hematopoietic stem cells, co-transduced by both MLLF9 and zinc finger-1 protein, we extended the panel of genes or molecular pathways potentially involved in this type of leukemia. So, taken together, our data demonstrate a role of zinc finger-1 in maintaining the immature compartment of malignant hematopoiesis. Zinc finger-1 together MLLF9 fusion gene contributes to the development and or maintenance of ML by altering the gene expression landscape. Thanks for the attention.
Thank you, Emanuela. Thank you. Can you hear me? Can you hear me? No. Okay.
Yes, Claudia, we can hear you.
Okay.
But
can you hear me?
Yes, now yes. Better.
Okay, okay. I had the problem with the. Okay, can you hear me well?
Yeah.
Perfect.
Now, yes. Okay.
Perfect. Thank you very much. So, thank you for the presentation. Very interesting. Just, I don't know if I missed something. If you there is correlation with the of MLLAF9 with the chromosomal mutation in this leukemia.
It's described that MLLF9 can activate zinc finger-1 promoter, but there aren't evidence about promoter mutation in this type of leukemia.
Okay.
Okay.
So there are some questions for Emanuela Chiarella.